30 seconds.
30 seconds.
Do we need to look at the panelists, the participants, and make sure that my committee is here?
Yeah, that's a good point.
Well, there's 45 of them, so that seems like a lot. Oh, it's increasing quickly.
Oh. Good.
Okay, great. Committee member here, Jan. Bree here. That's important.
Dave's here.
Hi, Bree.
We thought you were gonna come in a onesie, but
yeah, I thought about it, but Integrative Biology puts the videos on YouTube, so it will be there for into perpetuity. So I sort of balance the you know, how much of myself do I bring to this versus what lives on the internet.
Fair enough.
Flying scroll onesie specifically, exactly.
Okay, I think we should get going. I still see lots of people arriving, but all they'll miss is you know, a few remarks from me, which is fine. So in the interest of giving Anne plenty of time, good morning, everybody, and welcome to Anne Devan-Song's defense seminar. And as you all know, of course, this is a massive rite of passage for graduate students. They get to celebrate the research that they've been doing for the past several years with their colleagues and mentors and friends and family, and bring it all out in the public. It's a rite of passage, as it were, similar to maybe emerging from their chrysalis, or in the case of herpetologically inclined people, maybe rather hopping out of that pond, bright colors on display. What you might be less aware of is that this is also a massive treat for the students' mentors, of course. This is the one time when we get to brag in public about the amazing students who we've been working with for several years and who we've been hopefully helping achieve this growth that they're looking for in a graduate degree. And so brag I will. The format today, though, is that I share the bragging with James Watson, since we've been both co-advising Anne for the past 3 or so years. And helping her make network theoretical analyses her own, and that's really James's side there, and then apply them to a bunch of different data sets about animal societies and disease transmission. That's probably more me, and then wildlife trade as well, and also scientists, actually. But most of that Anne will talk about to you herself, and so I was going to focus my comments on some pieces that are not in the dissertation seminar here, because the dissertation seminar, of course, is a really incomplete representation of what a student has been up to and what they're all about. And so in Anne's case, the first thing that I'd really like to bring to your attention is that she is the most incredible field biologist. And I had the great pleasure of actually being in the field on one occasion with Anne in Costa Rica and going on a frog and snake walk, and witnessing firsthand what she can do in the field. And you would not believe how fast this woman will move, faster than that frog can say ribbit for sure, to dash and grab it, but gently, to then examine it and say hi to it, maybe take some really nice pictures of it. And perhaps slightly more questionable is the practice of Anne and her snake stick. Now, snake stick you'd think would be a defensive item that, you know, maybe you can rustle the vegetation to make sure that snake goes away before you get there. And that could happen maybe sometimes, but in Anne's case, it's more likely that she'll use that snake stick when she spots a snake and goes, yay, rather than jumping backwards like the rest of us, to pin that snake down and grab it and also say hi to it and take some nice pictures of it and identify it and see whether it's well and happy, interview it. So Anne in the field is a force of nature. And you'll see that reflected in what she's done as well in terms of her science and her publications. So Anne came to us from initially the National University of Singapore, where she'd done her undergraduate and worked on reticulated pythons. And I hope that somebody other than me spotted that, in fact, it's a reticulated python that foreshadows Anne's fate of becoming a network researcher already as an undergraduate. So pythons frequent the sewers and the sort of underworld of Singapore, and eat rats, which is great, but also eat pets, which is maybe not so great. And so Anne did some research on the habits of these creatures, which she published as well in Biotropica. in 2017. She then went on to the University of Rhode Island for her master's degree, continuing work on snakes, in this case a venomous snake, a pit viper, where she looked at their reproduction and at the practice of translocating them to get them out of people's backyards, essentially, but found that wasn't really the best option for snake conservation purposes. And she published 2 papers out of that as well. She then carried on with the collaborations she developed in Rhode Island and worked for the National Park Service in conjunction with her Rhode Island mentors on a toad species. And some of that will actually pop up in today's defense seminar, because she analyzed that data set on spadefoot toads as part of her PhD. So Anne's been incredibly productive with field-based research and publications. She's got, if you count in the ones that are accepted but not yet out, she's got 10 under her belt there, plus 4 natural history notes, which is just incredible. And I wish I'd been that productive that early on. And those adventures have taken her not only to Rhode Island and the national park in the eastern US, but also Luzon Island in the Philippines, and Mount Kinabalu in Malaysia, and South China, and of course her native Singapore. So, and then that little field trip to Costa Rica, lots of incredible field work. The other piece that I wanted to mention that Anne won't be talking about in her defense seminar today is that she's been really very active in diversity and inclusion work in the STEM field. And that comes out in committee memberships, of course, but actually there's also 2 chapters in her PhD where she talks about networks of scientists and how that influences their success, and how different measures of success might be more appropriate than your typical just numbers of publications and citation records that take more into account the impact that somebody has on their collaborators as well as on their own career. And then another piece on institutional transformation, basically, where Anne thinks about institutions in a framework of network theory and comes to some interesting new ideas and new conclusions there. And so that work is, probably equally as important as her science to Anne. And the way I like to think about it, and Anne too actually admonishes that committees are well and good, but it's actually what you do that makes the real difference. And for Anne, this is real. This is not hot air. And I find that when you think about the impact of a person on their environment and on transformation, of course, working in an organized setting is important because it's energetic individuals that breathe life into any kind of transformation. But really at the very base of it is how you interact with those around you. And so from the perspective of the Jolles Beecheler Lab, I would just like to say that it's been such a blessing to have Anne around in the lab, because she really does put her actions where she puts her words. So, whenever there is a new student, arriving from a far-flung place, Anne will be the first one to reach out to them and make them welcome and answer all their questions and take them around and help them settle in. Whenever there's somebody in the lab who's just stuck in the doldrums somehow and has a writer's block and needs somebody to help them make a plan and progress through that low spot, it's Anne who will help them with that and check in with them and make a time plan with them and make writing hours with them and help them succeed. And even when there's, a PI who's maybe pushed the brinksmanship of last minute grant writing too far and is sitting with a mess of unfilled references the day before submission, it'll be Anne that jumps in and helps out. And so this idea of people in networks helping each other out and helping each other succeed is something that Anne does. It's not something she just merely talks about. And so I think I can speak for all of us in the Jolles Beecheler Lab, and that we hope that these networks that we've built with Anne will be durable and that we'll get to collaborate and help each other succeed in the future as well. And so with that little bit of sappiness, I'd like to hand over to James.
Anna, thank you so much for that. I don't have the words to follow that up. I think everyone in me and everyone in the audience knows all of those words to be true. And so I'm just going to say, Anne, thank you. Thank you for being part of my lab, being part of Anna's lab, being part of our community, giving me the chance to work with you on the topics that you're going to present today. They are varied, and you've gone deep in each of them. And everyone in the audience, you'll see what amazing work Anne has done. And that's just the tip of the iceberg. There's so much more than the chapters in her dissertation. So Anne, thank you. And thank you for also just bringing your personality to our science. Often it can be, our work can be quite dry and dare I say a little boring, but working with you has been the opposite. So I'm greatly appreciative of having the opportunity to work with you. So with that, with my short little addition there, let's start the process. So I want to just describe to the audience, I think what most of us know is going to happen. So Anne will give her defense. This will be about 45 minutes. I want everyone in the audience, every single one of you, 78 of us, okay, to think of one question. All you need is one question to come up into your head, and I want you to write it down on a piece of paper, okay? It's very important. Don't forget it, because after 40 or 45 minutes, we have some time, 10, 15 minutes, to ask Anne ridiculously hard, impossibly difficult questions that she can't answer. And this is your chance. Okay, so please do write them down, put them in the Q&A. So there is a Q&A button. This is where you place your impossibly difficult questions. You have another option too. There is a chat. In the chat, you can heckle her. Try and distract her from giving her talk, okay? Do this. Enjoy that. This is an option for you. Importantly though, do get the questions down. After the questions, yes, we will leave and we'll go to a closed session with the committee where Anne will have a discussion about her work, and after that she can come back to you with the good news. Not getting ahead of myself, but because actually before we start, I have to read a poem. I'm sorry everyone if you're not familiar with this, but every defense that I'm partly in charge of, I feel like I have to read a poem. So Anne, this is your poem from the C Lab. Okay, and then after the poem, you may get going. So everyone, this is Toad by Mary Oliver, good old Mary Oliver, always a safe option. Toad, I was walking by and he was sitting there. It was full morning, so the heat was heavy on his sand-colored head and his webbed feet. I squatted beside him at the edge of the path. He didn't move. I began to talk. I talked about summer and about time, the pleasures of eating, the terrors of the night, about this cup we call life, about happiness, and about how good it feels, the heat of the sun between the shoulder blades. He looked neither up nor down, which didn't necessarily mean he was afraid or asleep. I felt his energy stored under his tongue, perhaps, and behind his bulging eyes. I talked about how the world seems to me 5 feet tall, the blue sky all around my head. I said, I wondered how it seemed to him down there, intimate with the dust. He might have been Buddha, did not move, blink, or frown, nor a tear fell from those old gold-rimmed eyes as the refined anguish of language passed over him. Anne, see you on the other side.
Thank you so much. That was wonderful. I'm going to share my screen now and do a quick test to see if everyone can see. Let's see, desktop 2. Can everyone see this and the red laser pointer? No? Red laser pointer. Fantastic. Excellent. So, there we go. So I want to start off my defense with my dissertation acknowledgement, because I would not have started or finished my degree without collective input from many entities. Let me remove this first, the closed captioning. Oh, never mind, whatever. Okay. I haven't seen my parents since 2018 summer, so this presentation is for them. Thank you to my wonderful family for being so consistently ridiculous my whole life. I appreciate my funding sources for my PhD, and thank you also to Jennifer Middleton, who's an inspiration to everybody who believes in equal rights. I have two one-of-a-kind PhD advisors, Anna Jolis and James Watson, whom I refer to as SURF and TURF. Thank you for giving me a home in your labs, providing me with this warm, creative, intellectual space to explore what sort of scientist I want to be. In an environment where everyone is brilliant and successful, the two of you have distinguished yourselves by being kind and lovely people, and I hope to work with you in your labs for the rest of my career. Thank you to my PhD committee, Jan Medlock, Dave Lytle, Bree Beechler, whom I refer to as the Council of Elrond, and to my wonderful lab mates who have created such awesome environments to be a scientist and citizen of this world. I have so much admiration for all of you, and shout out to Ricardo for being there for me for the last stretch. I am super lucky to have mentors and collaborators outside of my OSU labs that have helped shape my science of the past decade, especially my master's advisor, Nancy Carricker, and my old lab mate, Marguerite Walden. Mark Phillips and Kin Han have often been the lone voices of reason in a chaotic world. Thank you to Mark for being both my maid of honor at my wedding as well as my workout coach. And I appreciate many of the faculty and staff of integrative biology who have supported me along the way. I adore my Dungeons and Dragons crew. You're such sweet and special people, and the past 4 years of my life have been made wonderful by so many other people at OSU, and talking science and life and cooking feasts for all of you has been the highlight of my time in Corvallis. Thank you in particular to Erin Abernethy, Sarah Hamilton, Caroline Glidden, and Christy Cruz for your presence in my life these past 2 years. My acknowledgement slides are basically a super long-winded way of saying that all my science and products are group and collaborative efforts, which is why I want to start off my presentation with a famous quote from one of my favorite poets, John Donne, which represents a recurring theme throughout my presentation. In his prose work, Devotion Upon Emergent Occasions, John Donne writes, "No man or no human is an island entire of itself. Every man is a piece of the continent, a part of the main. Any man's death diminishes me because I am involved in mankind." Ignoring the overuse of the word man here, this prose work is about the interconnectedness of humanity. And the past 2 years of global events have starkly highlighted how individual decisions and interactions between humans reverberate through physical and social networks with consequences at the local, regional, and planetary scale. We can see emergence, or simply put, phenomena that are greater than the sum of its parts when we read the news or look at what's going on outside our doors. Phenomena like collective social action, herd immunity, insurrection, demographic segregation in cities, climate change. These are all phenomena where decision-making and organizing occur at a different scale than where the repercussions manifest. Network theory provides us with a unified conceptual framework for social organization of humans and other animals at all levels and for all types of interactions. At its most basic, network consists of unique units or nodes, where two nodes can be linked to each other through a predetermined definition of some type of connection, an edge. And a dyad consists of two or more nodes that are linked by an edge that can have some sort of weight or measure of magnitude. An example would be how many times you share each other's posts on social media. From this relatively simple definition of a dyad, networks consisting of many interconnected dyads can be examined at the node, community, and network level. And this framework allows for us to link individual behavior and decentralized interactions up to population level properties, including emergent properties of the group. Which brings me to the title of my dissertation, Emergent Properties of Animal Social Networks, 5 Stories in 5 Systems. My long-suffering committee will have to read all 7 chapters of my dissertation, but you, the lucky audience, will be guided through the main chapters, which form the meat and potatoes, or the bread and butter, or the rice and curry of my work, if you will. These 3 parts start with a story about a frog network, continue with a story about buffalo networks, and then end with global wildlife trade networks, and you will have friendly little icons guiding you through the different sections. And so begins part 1. Locations of dried up ephemeral pools explain assortative mixing of a fossorial frog. Or, if you wanted to John Donne up this title, no frog is an island entire of itself. So I stumbled upon this project purely by accident. I was conducting surveys for national parks in Virginia. And when I did surveys at night, I would get these overwhelming numbers of eastern spadefoots, this very, very cool subterranean frog. But not only that, I noticed I was getting clusters based on sex and size on non-breeding nights. We do expect adult frogs to congregate when mating, obviously, but I was seeing these patterns outside of any breeding season. I could find bachelor parties in one area with lots of beefy, sexually mature males. There was a bachelorette party in another area where the frogs were with child and getting ready to release their eggs. In another cluster where little froglets were hanging out like preschoolers. What I was observing with my eyeballs was homophily or assortative mixing, where like hangs out with like. This is super interesting because frogs are considered to be solitary outside of breeding aggregates. The eastern spadefoot is a terrestrial or fossorial frog that has this explosive breeding strategy for reproduction. Reproduction is triggered by torrential rains where ephemeral pools fill up with several inches of water. This picture here depicts a spadefoot pool that's filled up, and the next picture is a dried up pool, and all you can see is a dry field. Between 0 and a few nights a year, torrential rains fill up these dried up pools and trigger mass migration of spadefoots from their forested habitat to these newly formed ponds. I spent far too long on this animation, sophisticated animation for any of you to miss it. So here it is again, forest to pond. Breeding adults mate, they lay eggs in these ephemeral pools, and tadpoles hatch and develop incredibly quickly. And the ecology and evolution of the species is tightly linked to the extreme ephemerality of these pools. So I dug into the literature to see what I could find. We know that these frogs need specific types of soil to burrow in. Soil type dictates occupancy, which is presence-absence, but there's nothing in the literature that quantifies assortative mixing, and there is no explanation for this pattern. So, my research questions for this chapter are, is there assortative mixing in the spatial proximity of Scaphiopetes holbrookii? And 2, does the spatial arrangement of dried up wetlands explain this assortative mixing? So we conducted this research in Yorktown, Virginia, USA in the spring of 2017. We surveyed the shaded purple areas of the map, which follows the contours of the road. And you can see the dark purple spots on the map, which are the locations of all known dried up breeding pools in that area. 2 researchers walked on opposite sides of an asphalt road and surveyed into the forest using eye shines of frogs sitting on the leaf litter. Every time we detected a frog, we would step off the road, catch it, and record the sex, length, GPS location. And we have males, females, and SNBA, which is subadults and non-breeding adults. What I end up with is a bunch of points within the surveyed polygon, with each point representing a frog location with associated data. So I'm interested to know if there is assortativity or homophily, which is defined as a tendency to associate within proximity of individuals that are similar to yourself. I used a simple proximity defined network. Basically, each node is an individual frog. One frog is connected to another frog if both are within 25 meters of each other, which I can extract from the GPS points. From these networks, I then calculated the level of assortativity by size, by sex, and by size within these demographic categories. I will now use arbitrary blue and orange frogs to illustrate the Monte Carlo methods I used for the next 2 analyses. Got rid of that. All right, I can ask the question, are the orange frogs more likely to be nearer the pond than the blue frogs? And are the larger frogs closer to the pond than the small frogs? Monte Carlo methods involve comparing my empirical data that I collected with thousands of randomly generated data sets. Comparing the left and the right images, we can move incrementally in concentric circles away from the pond and ask the question, does my empirical data have more frogs closer to the pond than expected at random? Using the exact same concept, we can generate thousands of data sets where the GPS location of the frog remains the same, but randomly assign it a size from the distribution of frog I obtained from the empirical data. We can then ask the question, in my empirical data set, are frogs closer to the pond larger in size than expected at random? I performed these analyses on my actual frog data with multiple breeding pools and a more complicated polygon for survey area. And now let's move on to the results. First, is there assortative mixing? On the left graph, we have frog categories of interest and their assortativity on the y-axis. An assortativity of 0 means that there's no assortment, it's just random. 1 means that there is extreme sorting by type, and -1 means that they are disassorted, that they like to hang out with different phenotypes. The answer, yes, there is. Eastern spadefoots congregate by sex, by size, and by size within demographic categories. If we visualize the relationship between groups as correlation plots, with red interpreted as avoidance and blue as affinity, we find that subadults cluster together and avoid hanging out with adults, and vice versa, just like humans. So let's look at these three plots representing female, male, and subadults. On the x-axis, we have distance from the nearest wetland or breeding pool. And on the y-axis, we have the cumulative number of frogs within that particular distance to the pool. The black lines are my empirical data, and the gray lines represent the thousands of randomly generated data, and parts of the graph are shaded in pink if my empirical data is more extreme than 95% of my simulated datasets. Let's look at the females. For almost every distance between 0 to about 600 meters away from the pond, there are more females at X meters of a pond than expected at random. The same goes for males right in the middle. The black curve is consistently above this gray curve. We see the opposite trend for juveniles between 0 to about 380 meters away from a pool. We have fewer juveniles than expected at random. I didn't see any patterns with size assortativity except for males for about 40 to 180 meters from the pond, where we have larger male frogs closer to the dried up ponds than expected at random. So I hypothesized that the big beefy male frogs defend their territory at the prime real estate, which is near the dried up pools, beachfront property, if you will, and the early male frog that reaches a newly formed pool has a mating advantage. So that's all well and good, but what if I got lucky during those few nights of survey in 2017? I repeated the same analyses using a larger data set from 2016, which has repeated surveys of some shorter segments of the study area. And I noticed the same pattern with assortative mixing, corroborating that there is temporal preservation of these patterns even when the ponds are dried up. So in this chapter, I found that Eastern spadefoots assort by size and by life history category. I also found that spatial arrangement of dried up pools on the landscape explain assortativity by life history category. This means that dried up pools maintain aspects of frog spatial structure on the landscape, and there are two conservation implications for this. First, there is potential for locating unknown pools using aggregates of large male frogs, which is useful for wetland conservation. And second, we may need to consider existing guidelines for buffer zone protection surrounding wetlands, or reconsider them rather, which are often based on adult amphibians only. Basically, the ecological footprint of ephemeral pools far exceeds their size and their hydro period. This concludes part one of my presentation, and I will now leapfrog from frog networks to buffalo networks. Tough crowd, tough crowd. Part two. Relationships between contact heterogeneity and infection risk are best understood within the context of co-infection. Or if you want to John Donne up this, no buffalo is an island entire of itself. So, the fundamental phenomenon that I'm interested in for this chapter is the fact that social organisms experience variation in pathogen infection risk. If I had given this presentation in 2019, it would have started off a bit differently, but with the past 2 years of us living with COVID as a globe, we can understand what variation in infection risk looks like played out right in front of our eyes. We can ask ourselves two questions. Why did some of us get COVID and some of us did not? And what's the relationship between my exposure to an infected person and the risk that I get infected with COVID? Let's look at a fundamental prerequisite of infection with an infectious disease, which is exposure to the pathogen. In many disease transmission models, our understanding of infection risk is the exposure rate to the pathogen multiplied by some function of the likelihood of infection given an exposure. In the real world, however, few things are that simple, and that pesky part, the likelihood of infection given an exposure, is driven by a myriad of other complicated variables, including host physiology, external factors, and pre-existing conditions such as co-infection. The relative roles of variables 1 through 4 in driving infection risk is poorly understood, especially in non-human animals. And one of the barriers is the rarity of our empirical data that are equipped to deal with these types of complicated and multi-dimensional systems and questions. African buffalo present a highly useful model species to examine some of the questions that I posed earlier for multiple reasons. They are a highly social herd species with demographic differences in social contact. They're super wormy, lots of pathogens, many of which are sublethal, so your study subjects live on to be studied. And there are lots of diagnostic tools because it's a well-studied system. These pathogens represent a broad spectrum of parasite traits, including mode of life, transmission style, length of infection, and much more. So, my research questions. First, what definitions and metrics of contact networks are relevant for disease transmission in gregarious or social species? 2, what are the relative roles of contact heterogeneity, intrinsic host heterogeneity, co-infection, and the environment in shaping infection risk? And finally, how do the above two answers vary with pathogen traits? Let's look at the summary of the field data, which were collected by the Jolis Lab and Associates between 2014 and 2017. A herd of buffalo were kept in a large enclosure with the vast majority of the herd fitted with proximity collars, which measures their pairwise contact rate with each other. This herd was captured every 2 to 3 months over 3 years for a total of 15 observation periods that had usable contact data. We can think of the collected data falling under 2 broad categories. One group are our variables of interest, which is the presence or absence of 6 focal respiratory pathogens, 4 viruses and 2 bacteria, and our covariates or explanatory variables, which include contact metrics from the collars. You can think of it as who hangs out with whomst, host traits and physiology, and some sort of environmental variables. So let's take a closer look at the contact metrics used. If you take only one thing away from this presentation today, it's that sociality is multivariate and multidimensional. From the contact collar data, we can define the buffalo social networks by frequency, which is the number of unique contacts per unit time, or duration, proportion of time that 2 collars spent in close proximity with each other. For each network, we can measure the different aspects of each buffalo's position within the network, with each metric describing different behaviors. The strength centrality of each buffalo, for example, can be interpreted as a broad overall metric for how friendly the buffalo is. Edge weight variance measures how unevenly a buffalo spreads the love between its friends. And closeness centrality can be its Kevin Bacon index, where we play the game 6 degrees of Kevin Bacon. The lower the centrality score, the more connected it is to the rest of the herd. Finally, modularity is a network level metric of how strongly the network is divided into modules. So I used a group of models called conditional random fields models to estimate the relative effects of contact, host traits, environmental variables, and co-infection on infection risk. CRF models allow for interaction between dependent variables as well as variation in pairwise dependencies, in the case of our data, pairwise pathogen associations that can be influenced by covariates. The black lines indicate direct effects of a covariate on the occurrence of a pathogen. For example, pathogen C over here is directly affected by covariate 2. The red lines indicate a conditional association, that is, the relationship between pathogens A and C are modulated by covariate 3. I also calculated how well the model performed in predicting each pathogen occurrence individually. The first result is perhaps unsurprising, but needs to be said. Duration-defined networks are different from frequency-defined networks. All 4 of these networks represent the same time period using the same data, and on the left we have duration-defined networks, and on the right we have frequency-defined networks. In the row below, we have the same networks plotted into their respective cliques, which is related to the modularity analyses I mentioned before. The distribution of edge weights are different between these 2. And also the click assignment that each buffalo is assigned to is also different depending on the network definition. I therefore had to use both definitions of networks as covariates in my CRF models. Let's move on to look at results from the conditional random fields models. We can think of representing the pathogen interaction as a co-infection network where the strength and the sign of interactions are depicted as edges between each pathogen. We can see that the two acute viruses, para-influenza virus 3 and adenovirus 3, interact strongly and positively, which we can interpret as if a buffalo is infected with one of them, it is likely infected with the other as well. The opposite trend can be seen between bovine viral diarrheal virus, it's a mouthful, and the opportunistic bacteria Mannheimia hemolytica, where they interact negatively with each other. Now let's look at the results examining the relative importance of covariates in explaining pathogen infection risk. In this bar chart over here, we have our pathogens of interest, and the y-axis represents the relative importance of all the covariates in predicting pathogen occurrence. There's a lot of information, but I wanted to highlight an interesting and unexpected result, which was the conditional association between adenovirus 3 and Mannheimia hemolytica. Our results show that rather than directly influencing infection risk, closeness centrality, which is one of the contact covariates, acted upon the positive interaction between these two pathogens. And in turn, this conditional dependency was the best predictor of AD3 and MH risk. This result highlights the utility of conditional random fields models, allowing for interactions between your dependent variables. Other pathogens like bovine herpes virus were driven by environmental variables, while Mycoplasma bovis, the bacteria, was best predicted by host traits. Parainfluenza virus 3 and bovine viral diarrheal virus were both best predicted by associations with other pathogens or co-infection. But not all pathogens are equally predictable. And in yellow, I have area under curve, which you can interpret as model performance for each pathogen. 100% means full accuracy. 50% indicates your model does as well as a coin toss, which is useless. And we can see that the model performs pretty well for some of the pathogens, but pretty mediocre for herpes over here. And I would take the predictions of herpes with a grain of salt. A point to note is that of all the contact metrics I threw into the model, frequency-defined closeness centrality emerged as a predictor of infection risk. This means two things. How frequently you frolic with your buffalo compatriots is more important than how long each frolicking session lasts. And number two, a buffalo's closeness centrality, which is its Kevin Bacon index, how fast information spreads anywhere to that buffalo, is an important metric in infection risk. However, as I showed in the previous slide, it's important to note that closeness centrality doesn't directly affect infection risk, rather it modulates the strength of relationship between two viruses. So in this chapter, I found that a buffalo's position in a contact network, including its clique membership, depends on how you define the network. I found that the strength of some pathogen interactions are modulated by a buffalo's position in the contact network. I found that frequency-defined networks are more useful than duration-defined networks. And I found that a mixture of different host, environmental, and contact covariates explain infection risk, depending on the pathogen traits. Links between contact heterogeneity and infection risk are best understood within the context of co-infection. Basically, disease ecology needs community ecology. Here I conclude part 2 of my presentation and transition to more complicated networks where each node can represent collectives rather than individual animals. All right, part 3. Wildlife trade network structure across the tree of life is driven by taxonomy and economy. This is difficult to jot down up because no country is an island entire of itself is not a true statement. Instead, we can say functionally, almost no country is an island entire of itself, although geographically speaking, many countries are also islands, which really rolls off the tongue. Let's start by considering the scale and scope of the global wildlife trade. It's this huge industry, multi-billion-dollar global industry consisting of large-scale movements of wildlife within and between countries. This trade is not inherently bad. It supports the livelihoods of millions of people around the world. Unsustainable trade, however, is one of the largest contributors to global species decline through wildlife exploitation. The wildlife trade also facilitates the spread of diseases around the world, with implications for wildlife, humans, and livestock. Here are what I describe the four horse people of the unsustainable wildlife trade. There are data reporting issues for both legal and illegal trade. There is the illegal trade in itself, which is notoriously difficult to study and disrupt. There's taxonomic bias in research because people like cute and fluffy things. And there is a lack of tools to focus limited resources upon. These four horse people hinder effective use of available data for research, as well as for updating laws and enforcement of these laws. Much like any other global trade, global inequality drives the trade of wildlife across the world. Patterns in trade links, density, and volume change with socioeconomic inequality. For example, the total number of trade links decreases when the average GDP and per capita GDP increase, while trade links increase in quantity when there is a wider disparity in GDP and per capita GDP among countries. We can ask ourselves the question, why does the shape or the topology of trade networks matter? If we just go off sheer volume, it's pretty informative, but doesn't give us insights into the most cost-effective strategies for disrupting trade networks. I've illustrated three very different real-life trade networks using a coral, a turtle, and a pitcher plant example. In figure B, we have the coral trade network, where the nodes are countries and the links are trade volume between countries. The graph on the right shows how the network can be divided into separate modules, which in this case is just one module, modularity 0. The number of closed triangles trading with each other is 0, so transitivity is 0. Disrupting this trade would entail targeting the central node, in this case, the United States, but really politely, of course. In figure C, we have a more complicated turtle network where modularity is not high, but the number of closed triangles is high, and you have high variance in the volume of trade. One potential method is to disrupt this trade would be to target triangles with high volumes of trade. If we look at figure D, the pitcher plant trade has high modularity, which means it's strongly divided into modules, while it has a low number of closed triangles. So one potential method to disrupt this trade would be to target links that join different modules together while simultaneously targeting the central links within each module. So for this chapter, I'm looking for to quantify generalizable patterns in the legal trade networks that can be used to explain and predict network topology of wildlife trade networks across the tree of life. I'm looking for rules that transcend taxa. I'm also looking to identify beacon species in the wildlife trade that can shed light and act as appropriate subsets of model networks to study trade patterns. So let's start with a fundamental question. How do we summarize and characterize networks in a way that allows for comparison? With one metric, how can we quantify the difference in the serval trade network versus the ring-tailed lemur network? Here's where network portraits come into play. Network portraits are matrices that encode structural information about a network. Each cell in the portrait represents the number of nodes which have X nodes within Y degrees of separation from it. With one array, you can eyeball regularity and small world properties and many other useful summaries of a network that a network object may not be able to. More importantly, you can compare the distributions of these two arrays with each other. Basically, we want to know how different are these four portraits from each other, and I'm going to illustrate the concept with actual portraits. The Mona Lisa is a famous portrait that we may recognize instantly without having to describe characteristics of the portrait. And we can compare the Mona Lisa to other portraits and get an idea of how different they are from each other. All these paintings depict one woman sitting down with her hands folded in her lap. But I'm going to go out on a limb here and assume that you all agree with me that the top two paintings are more similar to each other than the bottom two paintings. The divergence of these two portraits is smaller than the divergence of these two. Let me know if you disagree. For every study year, we created directed and weighted networks for each species year combination, defining nodes as countries and edges as number of unique shipments per year. Every species year combination therefore had a graph or a network. We calculated the network portrait of each weighted network, which looked like barcodes or QR codes. We then calculated the portrait divergence between all pairwise combinations of species trade for each year between 1996 and 2017. Portrait divergence is an information theoretic approach to quantifying shape or topological divergence between two networks using a Jensen-Shannon distance metric. Portrait divergences provide a single numeric quantification of symmetrical dissimilarity between two graphs. This results in a diagonal divergence matrix for each study year. We use k-means clustering to assign group membership to each trade network within a year. K-means clustering tries to maximize differences between clusters and minimize differences within clusters. And I chose the top 3 optimal number of clusters for each divergence matrix. We can now go on to ask the question, what characteristics of a species drives group membership? And given a species with unknown trade, can we assign group membership? We used a regularized gradient boosting algorithm to conduct a multi-class classification of group membership based on portrait divergence. Gradient boosting is a supervised machine learning technique which creates an ensemble of weak decision trees that form a stronger prediction model using boosting. And boosting is learning from weak classifiers and iteratively adding them to a strong classifier. These models can then pass out the relative importance of each covariate in explaining cluster membership of wildlife trade networks. In order to identify beacon species, let's go back to the portrait divergence in the matrices from the previous example. From this, we can directly create a meta network or a network of networks, no relation to Facebook. In this definition of a network, each node consists of a species identity, and the edges between two species are how similar its network shape are to each other. In this illustration, the snake and tortoise networks have low divergence and high similarity, so thicker line. Our question is then, which of these species is the most well-connected or the most central species in this meta-network? Or in other words, what species is the best representative if you could only focus research on one of them? I defined beacon species as a highly connected node in this meta-network with a high eigenvector centrality score and therefore a great representation of this meta-network. I defined atypical species as nodes that were weakly connected, low eigenvector centrality scores that represented outliers in this meta-network. And let's move on to results. I have 22 years of data, thousands of species, nearly half of which are orchids. For every year, the thousands of networks could be optimally grouped into 2 clusters, which is not super informative. And the next 2 optimal level of clusters were between 4 and 21. Let's take a look at this graph, focusing just on the first panel. The x-axis is year, and the y-axis is the relative importance of each covariate in accurately predicting group membership of a network. Taxonomic covariates are in purples, economic covariables and covariates in green, and geographic variables in blue. There's a lot going on here, but the more you look at it, the more you get lost in dancing colors. But here are the main takeaways. First, taxonomy, particularly genus or taxonomic rank rather, overwhelmingly emerged as the best predictor of group classification via portrait divergence. This means that networks of species that were within the same genus were more likely to have the same network shape. For economic variables, Gini variance, the variance of Gini coefficient across all participating countries of a particular species trade, was the 3rd most important predictor of network classification after genus and family. Again, socioeconomic inequality drives the shape of global wildlife trade networks. Of all the geographic explanatory variables, the number of countries overlapping with the range of species had the most predictive power, but not that much compared to the rest of the model. And finally, conservation listings like CITES listings or IUCN listings had negligible importance in model predictions. All of the above patterns are fairly consistent across models, whether or not networks were classified into 2 or more groups per year. For the next results, the 50 beacon species and 50 atypical species represented a range of taxa from different geographic regions. Example of beacon species include this inquisitive-looking lemur, And examples of atypical species include the reticulated python, which is the world's longest snake and also one of the world's coolest species. Not biased at all. All right, let's take a look at the taxonomic groupings of the beacon and atypical species identification. Of the nearly 6,000 species traded, over half are orchids, and a fairly small percentage are reptiles, mammals, and birds. However, this breakdown is not similarly represented in beacon and atypical species. If you look at just the reptiles in dark blue, they represent 6% of traded species, but only 2% of beacon species, and 42% of atypical species. This means that reptiles are overrepresented as atypical species, and blanket rules and regulations for trade may not protect them as much. This is in contrast to birds, which comprise 9% of traded species, but nearly half of beacon species are birds. So in this chapter, I found that taxonomy and economy drive the structure of wildlife trade networks much more than geography or conservation. I found that reptiles are underrepresented and birds are overrepresented as beacon species. And there are two applications for this research. First, we can ask ourselves, given a particular species where trade is not fully known or reported, what is the most likely topology of its trade network? Second, given limited resources for disrupting trade networks, which species should we focus our attention upon? Our tools can be used to predict network structure of cryptic or unknown networks, and we can also have tools to focus research and resources on certain types of network. So if I could sum up my dissertation findings in one sentence, it's, I have more questions than I have answers. What am I doing? But through my different chapters, I've attempted to embrace some of these complexities and use multiple systems to ask and answer questions about the world that we live in. I used a common framework of network analysis to examine patterns and processes at very different scales. In part one, I started out with a static and fundamentally simple definition of networks, physical proximity, and asked what generates the spatial networks that we observe in a species of frog. In part two, I graduated to more complicated networks that represent contact heterogeneity to find out disease consequences of African buffalo social networks. In part three, I used and developed some tools in network analysis that allow us to extract data from the existing wildlife trade for the purpose of conservation. All of these chapters can be encapsulated in my dissertation haiku, which will empirically and unambiguously demonstrate why I pursued a career in biology and not poetry. My networks bring all the animals to the yard. Heck yeah, emergence. Finally, of all my OSU products, I am most proud of this one, which I am excited to launch post-PhD. Along with my colleagues, we are setting up the Beaver Dam Fund for minority retention, specifically for plugging the leaky pipeline in science by empowering women of color beeves. Stay tuned in the near future for more information and, of course, a website. Go Beeves! With that, I appreciate all of you for attending my defense. I hope that you have been adequately heckling me in the chat, and I look forward to audience questions in the Q&A that I am unable to answer. Thank you very much.
Applause. That was amazing, Anne. Thank you so much for that. That was such a wonderful presentation. Blown away. And I hope the audience, you realize the full scope of Anne's brilliance in each of these areas and the massive thing that she's drinking out of as well. Okay, all right. I asked you for this. Remember, the only stupid question is the one you don't ask. So let's go to some questions in the Q&A.
All right.
Can you do it, Anne, or do you want me to?
Well, I can see them, so I'm going to answer them live, and I can moderate it. It'd be great.
Yeah, and do read them out though, so we can all.
Great. So first question is from, I'm going to answer it live, from Zach Munier, my friend. Hello, Zach. Why might birds be overrepresented as beacon species in the global wildlife trade? So I actually don't know. I don't know why some species are over or underrepresented in the global wildlife trade. What the beacon species means is that many birds are similar to other, many bird networks are similar to other types of networks. And I don't know if there's something specific about the bird trade that makes it such and I would welcome any sort of interpretations of that. Maybe it's because they are traded in a particular way for pets or for for research and something, and while other species like reptiles may be traded for food or skin or things like that. So it could be due to use, but yeah, I'm slightly puzzled at that one. That was it's a good question. Okay, so next question is from my lab mate, Dr. Stephen Johnson. Regarding wildlife trade networks, given that taxonomy is a big driver of trade network topology, do you have any insights on what makes certain taxonomic groups trade-worthy? So I think it depends on use again. So it comes to what makes it trade-worthy. That's a question for what makes it trade-worthy within a particular use. So, what's ivory used for? It's used for decorative, you can make piano keys, or, it's for rich people to show off their wealth, as opposed to reticulated pythons, which are used for their skin, for leather, or even crocodiles. So what makes certain taxonomic groups trade-worthy is absolutely what the consumer end wants. And these are incredibly varied. It's all across the world. How people use wildlife is a myriad of like medicinal research, a whole pile of different uses. So that is a great question, but the answer would be specific to each type of use again. Thank you for the question. Okay, Jessie Laney. Hi, Jessie. Going back to your detected frog patterns chapter, what do you think are some of the drivers of assortative mixing you uncovered? Is there environmental filtering going on, or some interactions between groups, competitive exclusion, intra-species predation? Okay, so there's a lot of questions there. What do you think are the drivers of assortative mixing? So one big driver, of course, is the location of the pond. Where a male frog gets, the pool fills up and a male frog basically gets mating advantage when it's the early bird, basically gets the worm. So a frog has a mating advantage if it's right near the pool. So I would say, and the mechanism for it, which you're talking about, competitive exclusion or something, the mechanism within species is likely to be frogs defend their territory, their burrows or whatever, and the bigger male frogs obviously have a physical advantage, so they are able to get prime real estate at that area, which is near the pond. I don't really know what the answer is for assortative mixing in juveniles and females, however, because I didn't find a relationship between locations of ponds and assortative mixing in females, or inside assortativity of females or juveniles. So it could be a bunch of other things, including soil types. Certain frogs defend the territory better at more high quality soils. And there are other things like that. So intra-species predation, there are a few diurnal species that eat these frogs, which are nocturnal. So it's kind of interesting. I'm not really sure how they do it. So like you get colubers, or I forgot the common name, rat snakes, or racers that eat these frogs, which are usually underground during the day. So these diurnal snakes eat them. So I'm not really sure how that's related, but great question. Cameron Kehani, hi Cameron, my dungeon master. Is BVDV a hoax? Yes. All right. Anonymous attendee, why do you think genus mattered so much for trade network similarity? So yeah, this is a big portion of my discussion, which I hope to discuss with my committee. So I think, so there are two things. Firstly, genus and geography are related, right? So you have lots of endemic genera that are within the same area. So Madagascar will have lots of different genera that are, just unique to that island. But the other thing is that animals or wildlife within the same genus are more likely to have the same mode of life, or same size, or they're more similar to each other by definition, right? If they're within the same genus. So trapping methods or like collection methods might transcend that species level, and it might be easier to switch from one species to another. But those are my two hypotheses, and I'm super excited if people have more suggestions on how that works. Thank you, anonymous attendee. All right, so we have, how many more should I take, James? I think it's already close to the hour.
I want you to keep going for a few more minutes. This is good. I like listening to these. Yeah, so yeah, we'll keep going. Just answer maybe 3 more.
Okay, I'll just go in chronological order. Charlene Young, hi. Charlene's in Singapore now. What are the practical implications of your findings on herd and disease management for buffaloes or other bovids? That's an interesting question. I'm not really well versed in disease management, but I would say, so when it comes to co-infection, one of the interesting things is when you vaccinate, for example, a herd of buffalo or any sort of herd, right, for one disease, knowing the relationships between two pathogens would be super interesting, because do you vaccinate for both, the pathogens, or do you just do one and manage to control both? The other method besides, actual vaccination or treatment for managing disease in herds would be to separate modules. So I don't know whether my findings have great implications for the contact side of things, because our herd is super well connected. Every buffalo was connected to each other, and it wasn't highly modular. So that question could be answered with looking at multiple different herds which have more of a fission-fusion modular structure. More, James? All right. Thinking about trade and access to data, what are your thoughts on how your networks would change if you had more access to trade info in different languages? Very interesting. Okay, so that's a really difficult question to answer, but I can answer it in terms of my project, which is all my data are from CITES. So it's the international treaty. So all these different countries submit their reports to CITES for where they import and export wildlife. So I mean, CITES has a lot of problems, but the idea is that every single country that participates in it will contribute its data to that. That would be a really relevant question if you're thinking about within country or within region wildlife trade, and that might be wildly different. You have so many interesting trades that will that happen within a country that are not recorded, or at least not recorded in English. Great question.
Okay, all right. Let's stick to time. That's wonderful to hear, like how much detail you know in answering all these questions. There are a set of them that you didn't get to answer. I believe that'll be recorded, so maybe you can reach out to those folks who asked questions but you didn't have time to answer them to. Thank you, everyone, for being part of the audience. The heckling was fantastic. And yeah, those of you who asked questions, thank you again. That's great to see your engagement in Anne's work. And I'm sure Anne appreciates those questions too. So at this point, let's give Anne another round of applause and a big thank you. Really wonderful presentation. She will get back in touch with everyone in about an hour. Okay, so we're going to have a closed session with the committee, and then yeah, we'll move on from there. And the committee will log off and log on to the other link. Is that correct? Okay, so everyone. Thank you, Anne. See you in a bit.